Despite the development of very many new, very effective treatments that's continuing every single day, inevitably in all cancers, eventually the cancer cells find a way to escape the treatment. And understanding how they do that is an opportunity to identify new treatments. My name is Ultan McDermott. I'm Chief Scientist in Oncology at AstraZeneca, and I continue to work as a colorectal oncologist treating patients with colon cancer. Every Monday when I have my clinic, I see that although we have much better treatments than we had 10 years ago, in the metastatic setting, in many cases, we still don't cure cancer patients. And only if we understand the rules of how cancer cells are able to avoid treatment can we really design, I think, what are going to be tomorrow's new drug combination strategies. So the PERSIST-SEQ consortium is an exciting EU-funded collaboration which brings together industry and academia. And that in itself is quite special because many of these types of funded endeavours tend to be either academia or industry, but never both together. So there are two co-leads. I'm the industry lead from the AstraZeneca side, and Alexander van Utenorden from the Hubrecht Institute is the academic lead. We bring different strengths to addressing this important question around drug resistance. I've worked for AstraZeneca for approximately 7 years. And I bring the clinical experience of treating patients, but also the more cutting-edge scientific research involving single-cell sequencing, and in particular with a focus on understanding drug resistance. I'm based and I work here at what's called the DISC, which is AstraZeneca's Discovery Centre, right in the middle of Cambridge's biomedical campus. So we're surrounded by many of the top UK academic institutes and hospitals. And for AstraZeneca, it was important when we set the DISC up that we were part of that ecosystem with the understanding that no one company can do everything we need to do to improve the lives of cancer patients. And so working with academic collaborators, working with hospitals is for us critically important in how we bring our science to the point where we can make new drugs for cancer patients. Because it's only through partnership and collaboration we can really, I think, develop tomorrow's new exciting cancer drugs for patients. AstraZeneca's motivation for co-leading the PERSIST-SEQ consortium is the realization that despite all of the best therapies that we and other companies are developing, the reality for most patients with advanced or metastatic disease is that we do not cure those patients. And so it became very clear to us that as we begin to think about how we combine drugs together to hopefully eventually cure patients, part of that drug combination strategy has to be underpinned by a better understanding of how cancer cells survive treatment. Part of my role is working with my academic colleague Alexander van Utenorden in Utrecht, where we both coordinate what are the types of cancers we want to apply quite a cutting-edge technology called single-cell sequencing to try to understand why cancer cells are able to evade or avoid drug treatment and survive drug treatment. The simple reality is that cancers are very good at finding a way to escape the effects of treatment. And we don't really fully understand how cancer cells are able to do that. So fundamentally within the consortium, we generate single-cell sequencing data from a whole range of different models. And each of these models is analysed in the absence or presence of a particular drug treatment. And that drug treatment could be a hormone treatment, it could be a chemotherapy, it could be radiation, it could be any treatment that we use in the clinic or planning to use in the clinic. And what we're looking at is understanding that whenever we apply a treatment to a cancer model and we look at the cells that are still alive at the end of that treatment, understanding why those cells are still alive and understanding what's the biology of those cells is really the core mission. We hope that'll give us clues into what the new drugs we should be developing to target what we call persister cells. And we call these persister cells because these are the cells that persist despite treatment. And the biology of these cells is very poorly understood. So this is a super hot topic globally in beginning to understand how we design better drugs to treat cancer. Bringing together both academia and industry, I think, has been critical for the success of the PERSIST-SEQ consortium. We bring different expertise, but also different viewpoints. Some of our collaborators have huge expertise in mouse models of cancer. Some of our collaborators have huge expertise in what we call organoids, which are a type of next generation cancer model. Some of our collaborators have access to patient samples, and it's only by bringing all of those collaborators together we capture the breadth of different models we need to adequately address the question of what's the biology of persister cells and how do they survive treatment. Given many of these groups, we live and work in different countries, there's an element of coordinating how we work together. And a key foundation of the PERSIST-SEQ consortium is that although Each individual group generates its own data. Everybody sees everyone's data. So it's a shared data resource, and that's very important. Alexander has very deep expertise in the technology side of single-cell sequencing, and what I bring to the table is much more the clinical and pharmaceutical expertise around drug discovery. But what we make sure, I think, ultimately is that we're going to deliver to the goal of the consortium, which is at the end of the 5 years, we have analysed a sufficient amount of single-cell sequence data from a range of different cancer models, such that we can begin to identify what tomorrow's drug targets should be to try to eliminate these surviving cancer cells. So we spent the first about a year and a half developing a lot of what we call protocols, because this is quite a cutting-edge technology. And so for the last probably year and a half, we've actually been doing sequencing. And I think an important milestone for us was when we passed having sequenced more than 1 million single cells. So at the moment, we've sequenced about 1.5 million single cells. A large number of those are the persister cells, and that's across a range of lung and colon cancer models, but increasingly beginning to bring in some breast cancer models as well. Another milestone was moving from the very simple models, which is cancer cell lines. So these are cancer cells grown on a dish, on a bench. Been able to move from generating data in cancer cell lines to generating data in what we call organoids. They're considered to be more patient relevant. Moving from organoids then into the much more complex mouse models where you can grow cancer cells in a mouse and treat in that setting. We've done that as well. And now we're moving towards getting data now from patient samples where we can actually sequence samples from patients before and after treatment. No single model, whether it's a cell line or an organoid or a mouse or a patient, no single model gives you the entire story. It's only by analysing across all of the models you begin to get the real signal as to why persister cells survive treatment. SEQ is focusing on a concept we call tumour plasticity. What we realise is that in addition to mutations driving resistance, is that there are other ways cancer cells can become resistant. And these are through non-genetic mechanisms. We don't understand the rules of that. But the first step in understanding that is beginning to collect the data we're collecting in PERSIST-SEQ. We're looking at cancer models before treatment and on treatment. The ultimate goal is to translate that understanding into tomorrow's new drug targets. That for us is the most exciting element. That's how we help patients. The reality is that for every new drug we develop, this is the problem we encounter for every single patient, which is that drugs work very well in the beginning, but then cancer cells find a way to turn certain programs on or off to survive treatment. And what we need to develop is essentially an encyclopedia or a knowledge bank of what those programs are that cancer cells do use. And by understanding that, to identify new drug targets. Another certainly benefit of this consortium is to develop and explore new technologies. So at the moment we've begun to talk about single cell sequencing. What you do not capture is whether cell A is the neighbour of cell B, or cell A and B are completely separated. And that's important because we know that cells signal and talk to each other. You can only capture that by looking at spatial transcriptomics. So it's an important new technology that we now have access to in 2025 within the consortium. It's not instead of single cell sequencing, it's something you would do in addition to single cell sequencing. It is very clear that there are questions which we do address within the consortium which may feel to some individuals has been more academic, but we're still at the stage of understanding the biology of persister cells because we think ultimately that they will over time still give us insights into what's going to be clinically relevant. One way to anchor the PERSIST-SEQ consortium in what's relevant to patients is focusing on cancers which are highly prevalent and where we really need much better treatments. And so for that reason, the PERSIST-SEQ consortium is focused primarily on colorectal, lung and breast cancer. Those are cancers which affect many patients and families. We tend to focus on common cancers and we try to focus on drugs which are in the clinic today or will be in the clinic in the near future to make discoveries that are clinically relevant. As a pharmaceutical company, we cannot do this on our own. And therefore the rationale for the consortium to bring together both academic and industrial expertise, to bring together pharmaceutical companies like Bayer and Merck, and to bring together academic institutes, Hubrecht, the Wellcome Sanger Institute, IRB in Barcelona. There's no one single institute or company can do this by itself.